Thank you so much for the kind introduction. It is really, really a great honor to present today a little bit about our research that we are doing in small cell lung cancer and really trying to get a little bit more precision medicine in this cancer type. So I'm really, really honored that I have the possibility to speak about this today and to share a little bit about what we are doing. I think I don't need to give much of an introduction about small cell lung cancer in this group. This became really clear. But the important thing is that small cell lung cancer is still the minority of the normal lung cancer cases that we have. And I have the impression it was sometimes a little bit a good excuse to not give it the kind of, importance and the kind of discussions that is needed, because when we speak about lung cancer, we always kind of fall back to non-small cell lung cancer and forget that there's also, small cell lung cancer, which needs to have a lot of attention. And therefore, I'm very happy to share a little bit new research news about this specific cancer type. The important thing is when we now look at the lung cancer, and especially non-small cell lung cancer versus small cell lung cancer, then we can really see that for small cell lung cancer, there's a lot of things which have been going on in the past years. There was a lot of, actually the whole field was exploding. There were a lot of, new research, a lot of innovative therapies that have been developed, a lot of different targets. While for small cell lung cancer, we unfortunately, at least for the data we have till 2016, it still feels like there's not much that has been really done to improve the therapies that we have for the specific cancer type. For example, just to show you this, just in 2021, there were like 246. I was counting it before, now I forget. There were like 6 drug approvals just in advanced non-small cell lung cancer. This year in 2022, we had another 7 drug approvals in non-small cell lung cancer. I think the last one was last week when there was the second KRAS inhibitor, which was approved. And we've really seen in non-small cell lung cancer that all those new drugs which have been developed in all different lines of therapies. Unfortunately, for small cell lung cancer, we can actually get 40 years of drug development on one slide. So it is really, really clear that all those new advances that we have seen still yet need to be introduced in small cell lung cancer. And there were a lot of positive for development. And really the newest development that we've seen in small cell lung cancer was mostly the introduction of immunotherapies that have been developed in the past few years and that really started to shape how small cell lung cancer is treated. But even there, when we look now at those new therapies, actually 2 of those have already lost their approval by the FDA because they were not proving to be sufficiently efficacious that was expected in the beginning. So in small cell lung cancer, we will really still need to develop much more therapies. We need to be much more better to find new ways how we can treat this cancer. The probably biggest advancement in probably 20 to 30 years was then really, or the issue that persists is still, when we wanted to develop those new therapies, we really have to know how can we hit the right target. And hitting the right target in small cell lung cancer is really still a big challenge because we really need to find the right targets that we can hit. And the moment that we have those targets, then we can really start to develop the right therapies. And the problem that we have actually with this is that when we look now at the genomic profile of small cell lung cancer, when we look which are the genes that are different, that have been mutated, that have been changed in small cell lung cancer, it is actually quite eye-striking. This was a paper which came out like 7 years ago already and clearly highlighted that all of those, or almost all of the small cell lung cancer cases really have mutations in 2 genes that we call TP53, and they're lost in another gene that we call RB1. And this is kind of the hallmark of all small cell lung cancer cases. The important thing outside of this very, very pronounced and very prevalent genomic alterations that we see in small cell lung cancer, however, and this has become even clearer now, much bigger studies that we have, there are virtually no what we call actionable mutations. So they're not mutations that we know, non-small cell lung cancer, for example, where we hear about EGFR mutations, we hear about ALK fusions. We have heard a lot now, for example, recently KRAS mutations. So all those mutations, which are non-small cell lung cancer, make up a huge proportion actually of the tumors. And where we have managed to develop drugs for these cancers, we unfortunately cannot find them right now in small cell lung cancer. The important thing is, so really the biggest advancement that we had in the therapy of small cell lung cancer over the past 20, 30 years was really the introduction of immunotherapy. So the first study actually demonstrating that immunotherapy can, when added to a platinum and etoposide chemotherapy, can really improve the therapy of small cell lung cancer was a study. It's called Empower 133. I'm saying this because I will come back to that study later, which demonstrated that atezolizumab, which is a antibody blocking PD-L1 molecule, was really able to improve the overall survival in small cell lung cancer. And today we also have a second drug, which is durvalumab, which in another trial, which is called Caspian, also showed that this is really a drug also targeting PD-L1 that can really improve the therapy in small cell lung cancer. The important thing when we speak usually these days about cancer therapies, we speak a lot about biomarkers. How can we select the right therapy for the right patients? How can we do this in the appropriate way? And there have been a lot of new assays which have been developed to really guide this. The important thing for the addition of immunotherapy in small cell lung cancer is that this is actually not guided by, so to say, biological biomarkers, but this is really a therapy that should be given or based on the guidelines for most of the patients. Obviously, there are a lot of clinical parameters which guide the therapy decision, but it's not that there is a specific biomarker like in EGFR, that you have an EGFR mutation, you get this EGFR therapy, but it's really given to most of the patients. And whenever we have the situation that we have a drug that was developed in this way, we're usually asking us, so how can we figure out who will benefit the most of the therapy? So how can we understand, especially when we have this, let's understand what is going on and really is there anything that helps us to define which patient has the best benefit of this therapy, or can we then, for example, define an alternative therapeutic strategy? And the important thing when we were speaking about small cell lung cancer is we are used to do this usually with looking at mutations, if we see some alterations of the genome that guides it. But we know already in small cell lung cancer that this is something that's actually most probably not working because we don't see those pronounced genomic alterations that can really help us to guide this. So what we were actually doing was thinking about what are the other possibilities to look at. And just to give you a very, very basic, this is not supposed to be a lecture in basic molecular biology, but the important thing to understand is when we speak about our genetic code and how molecular biology is always working, we usually have this idea that we go from the DNA, which stores the genetic information, in a process that we call transcription, to building RNA. And this RNA in a second process, which we call translation, is then building the protein. And in a very, very simplification, it is much, much more complex than this. Usually it's the protein which is building out the function, but we have this multi-step process to go there. The important thing is, so now we know, okay, we cannot look at DNA, or we can look at DNA, but we will not find anything. So what are the other possibilities that we can look at? And the general thing is when we look at proteins, it's a little bit more challenging to look at a lot of different proteins because the methods are a little bit more complicated. We can do it, but it's not always that easy. Well, we usually like to look a lot of RNA when we want to learn more about the function because it's something that is very easily analyzed, and while there are certain nuances certainly to it, usually the idea we see of a certain RNA that is produced of the subsequent protein we have, and therefore we get kind of an information on what is the function that is happening in a certain tumor state. The important thing is those programs are controlled by proteins that we call transcription factors. So transcription factors are basic proteins whose role is to really change the transcription, so this production of RNA, for a lot of different targets. So it's not just changing one, but those transcription factors are really, especially in development, capable of saying, now we make an arm, now we generate a brain. So they are really proteins that are capable of orchestrating a lot of those different fine-tuning and changes that really allows us to actually build our body. And the reason why I'm speaking and telling you all about this, about transcription factors, is actually that it has been clear over the past decades already that there are certain very specific transcription factors which seem to be very different in small cell lung cancer. And here you can also see how we really have sometimes stepwise approaches that really started in the 1980s, where we had kind of the first starting to classify small cell lung cancer. Like I say, finding the right target. We like to make little buckets and then, in the little bucket, understand what is going on. And the more we can do it, the better. And especially in the past years, it's not even 10 years ago, it became kind of clear that there are certain of those transcription factors, so those really strongly fine-tuning proteins that are associated with certain types of small cell lung cancer. The first that we have been described as ACL1 and NeuroD1, and I will come to them later. And what happened now over the past years, and you can really see also how the research accelerated from 30 years of paucity in research to really seeing how things are going and evolving very quickly, it became absolutely clear that while we do not see those genomic changes in small cell lung cancer, we see a lot of those transcriptional changes, we call it, where we have those big changes in the RNA composition, changes in different proteins that really make differences how small cell lung cancer look like. And the important thing is there have been 3 very important transcription factors which have always been rediscovered and always came back to be able to play an important role, which is ACL1, NeuroD1, and Tau2F3. The important thing is we sometimes tend to, while we like to put things in buckets, we sometimes, get a little bit too stuck in our buckets, and then we are, pre-building buckets, and then we always want to fit something inside, and this is also sometimes a problem. So what we were actually trying to do, we call this an unbiased approach, and we actually took a step back, and there are now very complicated machine learning approaches. I will try to speak as little about machine learning as possible, but I will tell you a little bit about. But the important thing is there are computational algorithms which are actually capable of telling us how many buckets we have and what are those buckets. So it was not us thinking how can we redesign small cell lung cancer, but actually we let the data that we had trying to redesign how can we actually classify small cell lung cancer. And what we did, so we used one of those methods, which is non-negative metric factorization, only if you happen to work in mathematics. Frankly, even I don't understand how precise it is working. But the important thing is, with this method, we figured out that it seems like 4 buckets is the right number. And then we selected 1,300 of those genes, so of those RNA transcripts that are then building a protein. And this is, why we usually like to work with RNA, because we can look at hundreds of thousands of different transcripts at the same time. And what we could really figure out was that we have those 4 buckets, and those 4 buckets have very specific phenotypes. So not only do we see buckets, but those buckets make biological sense, meaning that this can really allow us to better understand the biology of small cell lung cancer. And the important thing is what came back from something that we've seen is that 3 of those buckets are really, of those subtypes, are really defined by those transcription factors that I told you before, ACL1, NeuroD1, and Tau2F3. And therefore today we call the different small cell lung cancer subtypes even small cell A when it is defined by ACL1, small cell N when it is defined by NeuroD1, or small cell P when it is defined by Tau2F3 to make it a little bit easier to understand. The important thing is when we did this, we also saw that there was a 4th subtype where we do not see this transcription or the predominant transcription of one of those transcription factors, but we saw a very inflamed phenotype. And inflammation is something that is usually something meaning that you have a high activation of the immune system, and therefore we call it the small cell I or inflamed subtype. The important thing is we were now able to also define this, that we can really see that we see those transcription factors are really clearly defining the biology and those subtypes to really help us to define those 4 subtypes. The important thing is this is always something which is very interesting, and this is really helping us to understand the biology, understand what is going on, to then define therapies on it. But the most important thing is when we see something like this, is this therapeutically meaningful? Because in the end, we want to develop new therapies. We want to improve the outcome. We really want to find new targets, really hitting the right target in small cell lung cancer. So the question we always ask when we find something like this, can be associated with a certain therapy? Can it help us to develop something new? And the important thing is that we were actually very happy that we had access to this IMPAL 133 clinical trial. So the clinical trial that I was showing before would establish atezolizumab as a new therapy option in small cell lung cancer. And now we had the possibility to look at this big clinical trial and look at our subtypes, how we can actually understand if there are, like I said, are there groups of patients that are have a better response to the therapy. And indeed, the inflamed subtype, the I subtype, where we saw a higher activation of the immune system, was indeed the small cell lung cancer subtype, where we saw the best response to immunotherapy. So this is clearly highlighting to us that when we look at those subtypes and when we try to find new ways how we can define small cell lung cancer, that this can really help us to guide therapy or hopefully to define new therapeutic options. And I'm only showing you today the outcome about the small cell I or the inflamed subtype. But the important thing is that we also think that we found biological understanding or understand the chemo more, that also for the other subtypes, we have an idea what are new therapeutic strategies that we can try to develop. And it's really everybody in the small cell field who's doing research is really excited, because we always, everybody has an idea how we can now really try to define new therapeutic options. The important thing, however, is now we have defined this and really want to demonstrate that this is the way we should do it. This is how we can really move forward and run new clinical trials, develop new therapies. And the first thing that we actually had is, how can we classify the small cell lung cancer? Because, we made a research project there. We were looking at very good samples, but we should have the possibility that we make this classification for all small cell lung cancer specimens that we get, that every patient who gets a small cell lung cancer has the possibility to have the classification of their tumor and ultimately in the future, the right and appropriate treatment for this. And the first step that we were actually doing is that we were building a predictive classification system that allowed us to classify now small cell lung cancer specimen using this RNA, so this transcriptionally defined subtypes that we had there. And I will promise you, I will not speak a lot about machine learning, but the important thing is we are looking at a lot of data here. And whenever we are here using a lot of data, we need to make sense to it. And machine learning is a very important tool. Maybe you've seen recently the news. There was a new chat AI, for example, that draw a lot of attention. I was reading in the New York Times, for example, about it. Now everybody is using it to write emails or whatever. But the important thing is machine learning is a subset of a huge amount of different tools, and it might take hours to even speak about it. But it's really something that we use to make sense to data. And sometimes jokingly, when we don't know what to do, we use machine learning and it tells us what we can do with the data. And then we get new results and super nice publication and we are happy. In reality, it's a little bit more complicated, but I promise I will not go too much into detail. But I will just give you a very brief overview how we are trying now to make the classification of small cell lung cancer and something that we call consensus classification. Because one problem that we have with machine learning is that, to the best of my knowledge, all those algorithms are forced to give you an answer. So all those algorithms are forced to give you an answer. And to the best of my knowledge, they are not capable of giving you the answer, I don't know. And this is something which was extremely important for us, because those small cell lung cancer subtypes are new. We are really trying and starting to understand what is going on. And we want to ensure that what we are looking at is something that we really understand and that we have the possibility that we don't know. So what we were actually doing, we called this consensus approach, that we split now the whole small cell lung cancer into those 4 subtypes. And we built predictive models for each of those 4 subtypes individually. That we only have the possibility to say, is it small cell A? Yes or no. Is it small cell N? Yes or no. Is it small cell P? Yes or no. Because this then gave us, in the end, the possibility that we built this consensus. And we were saying when more than 50% of those models tell us this is an A, then we say it is an A. And when we do not reach this threshold that more than 50% of our predictive models go, we call this equivocal, which means we don't know what is the subtype. We are incapable of assigning the right subtype, which gives us a little bit more security that we are really ensuring that what we are looking is really there. The important thing is what you can see here is now actually the outcome of the consensus classification, which gave us a little bit more insight in the end that we were thinking. And I will try to guide you through it. Again, when you have questions, really never hesitate to reach out and interrupt me. I can tell you I've presented this multiple times to a lot of highly specialized cancer researchers, and even they needed 2 or 3 times. So really, there's no problem.
Oh, sure. So can we stop you for a moment to ask a few?
Absolutely.
Good chat. Okay, so one of them, and we have some good, pretty good ones. One said at the end, but I think we can ask it now. So they said for the father was given an immunotherapy drug, durvalumab, along with initial chemotherapy. He was diagnosed with type 1 diabetes, and they said it was because of the immunotherapy caused his body to attack the pancreas. Since then, the doctors say that he is not a candidate for other immunotherapy drugs because it could be dangerous. Any experience with a reaction like this, and if it really makes all types of immunotherapy not possible for him?
So the important thing is, regarding this question and related question, I'm not a treating physician. I'm a cancer researcher. I'm not a treating physician, and therefore I cannot give any comment, recommendations, or anything. I'm really trying for the research, so I'm very, very sorry that I cannot address those questions.
Oh, no problem. Well, we'll open it up to the forum later. Maybe somebody has an experience with that. Also, and I was interested in this too. Immunotherapy for small cell lung cancer. Did you say that there's only one type of immunotherapy now approved for small cell lung cancer? Wondering about the distinction that makes this work.
So there's, so to say, one type of immunotherapy in the frontline setting, which is blocking PD-L1. So this is the molecule. There are two molecules actually which are doing this. There's atezolizumab and there are durvalumab. So those are two distinct therapies, but they are both doing the same thing. They're both we call them antibodies. So those are specific kind of protein that is blocking a protein which is called PD-L1, and this is something that then helps us to release the power of the immune system. So there's like one type which is approved, but there are two drugs, atezolizumab and durvalumab, in the USA.
And what you're talking about now in your presentation, would this be the equivalent of a TKI, a non-small cell?
No. So the important thing when we speak about TKIs, those are very specific drugs which are targeting proteins. This is where it's coming from, kinases. Kinases are proteins that are, how to say, transferring signals inside of a cell. And those are extremely powerful because many of those signaling molecules are mutated. And when they are mutated, for example, in the case of EGFR, for example, they give a signal all the time. For example, a signal grow, cell grow. And when they are mutated, they always give the signal grow, grow, grow. And when we have a TKI, those are very specific drugs which are then capable of blocking this signal that this aberrant protein is giving of like growing. When we speak about immunotherapy, it's a relatively different concept. The concept is actually the drug is not attacking the tumor itself. The drug is reactivating the immune system, and the immune system itself is then attacking the tumor. So one thing is really like, we call those targeted therapies, because you really hit one target and you hit this specific mutation, which leads to sometimes spectacular results. While with the immunotherapy, it's a very innovative. I mean, it's there for like more than 10 years, but I still believe it is very innovative, where you have really the body itself is trying to get rid of the tumor, which is also working sometimes spectacularly well because you have much more cell lines and not just this one target. The important thing is, when you have a target, this is always good. In small cell lung cancer, we don't have those targets because those require those very specific mutations. Maybe we will find them and maybe we will be capable of developing them, but right now we unfortunately don't have them.
Right. Oh yeah, and I think that answers. We have two more. I think that kind of answered this question concerning the subtypes. Are there pipeline drugs to target subtypes? I understand that these subtypes are prognostic only at this point. Is that correct?
Exactly. So at this point, they are only prognostic. We've seen differences. All the work that we are actually currently doing is precisely this, proving that they are predictive, that we can develop drugs. And the important thing is, this is kind of the research that I'm now trying to we're working on it. The most important thing when we want to do it is our capability to diagnose those subtypes. Because we are extremely good in detecting mutations. Detecting mutations, yeah, of course, you always new ways, and there might sometimes be limitations, which are, due to more technical limitations or sometimes different things. But we are actually pretty good in detecting those cancer-related mutations. This is really working very well today. But now we are no longer looking at mutations. We look at transcriptional changes. And the important thing is, when we look at mutations, we look at a yes-no event. Is there a mutation? Is there no mutation? And this is, to a certain extent, much more easier because you only have this binary outcome. When we now look at those transcriptionally subtypes, we look at something is a little bit more than something else. And this is a much more complicated approach. And to the best of my knowledge, so everything we are really trying to do here will be a first, because to the best of my knowledge, there's not a single approved drug based on a transcriptionally defined cancer entity. So we are really here trying at the forefront to really actually revolutionize how we can do this. But sometimes the reason that something hasn't been done is because nobody had the idea, and sometimes the reason nobody has done it before it was because it wasn't possible before. And we are now really trying to solve more technical problems and to really get this. And this is, for example, this consensus classification. I will show you. This is just one of the steps that we are working on to really have the possibility to prove. Currently, everything I'm telling you, it's prognostic. We are very strongly convinced that we will have the right therapy for the right subtype, but we yet have to prove it. And proving relies on our capabilities of really classifying small cell lung cancer in a routine context. That every patient or every patient, it's not always possible to get enough, for example, tissue specimen or something, but that that we give our best possibility that most patients really can have this classification, that it's always the same when we look at it, and that we can then assign the right drug. So we are really on the forefront, but there's still a lot of work to do.
Yeah, and I can tell, I'm a witness that even this prognostic research is, in the last couple of years, this has boomed. So this is hopeful for us. And I think we have one more question in the chat. We'll turn it over to you. I think you've answered the first part of it. What are the implications of SCLC, AN1, et cetera? I think you're saying they are prognostic. And this is a good question because we know it happens in non-small cell. We always tell them to get their tumor tested. But how can we know if the subtype of our small cell lung cancer, and why does it matter?
So right now, that's the issue. You cannot, because we are still developing those diagnostic possibilities that we can do it. So everything we are doing is a complete research project. So those are things that we try to understand more. But I can tell you, we are giving our very best to really get those diagnostic methods very soon. Why it matters goes to the discussion that we had before, because hopefully, and again we have to prove it, but hopefully. We will have the possibility to give specific drugs for specific subtypes. And then we really try to enter this precision medicine in small cell lung cancer, that not every patient get, to a certain extent, obviously, the same drug, but that we can really say, like, okay, when you have this subtype, you get this drug. For example, when you have small cell I, oh yeah, then immunotherapy is the appropriate treatment. But when you have a small cell A, maybe there is another therapy which is more appropriate. And this is the idea that we can then really match the right therapy to it. Again, everything we do right now is pure research. We are, like Montessa just said, it's happening so quickly right now, and so many new things are working on. Many groups are in parallel, but it's still something that it's research right now.
Well, thank you so much, and we'll turn it back over to you. If anybody else has any questions, please put them in the chat, or you could use the reaction and raise your hand. This is very informational. Thank you.
Thank you very much. So now what we have, we're going back to the classification. So how can we classify it? We now have this consensus classification, as we call it. And actually, the more green, so each of those columns is one tumor sample. And what you can see is the more green you see in this respective column, for example, for small cell A, the more of this predictive models that we build is actually predicting that this is a subtype. So the more green we see, the more we are certain that we have a certain subtype. And then, and this is the interesting thing, we also looked at the expression, again, the amount of RNA molecules that we have for those 3 transcription factors, which I was mentioning in the beginning. And what we can really see is that the patients or the tumors that we have classified to be of a small cell A subtype are the ones which are strongly expressing this transcription factor that we define. And therefore, we are now very, very confident that what we're actually looking at is a very good and robust classification system. And importantly, we also have those 4 samples that we could not classify because we couldn't understand what is going on. And this is very important because, again, we don't want to give a wrong answer. We prefer having no answer than a wrong answer, that we can really get the right therapy to the right patient. The important thing, like I said, we are actually doing a first there. So this is something which has never been really achieved. So we are trying to build a lot of confidence to understand that what we're looking at makes sense and that we really maintain the biology that helps us to address the right therapy. And what we actually had the possibility is, so initially, this was a paper published by Dr. Carl Gay, where we had this first explanation and classification of those 4 subtypes. And we learned a lot how those subtypes are actually looking biologically. For example, we were looking at genes which we call neuroendocrine because they are associated with, a more neural phenotype. And there are genes which we call them non-neuroendocrine. It is just a distinction because we know that some of those subtypes have more this neuroendocrine phenotype, as we call it, and some of the subtypes have a different phenotype. And we were recreating now with our new cohort that we have created in-house, and we see basically the same that we've demonstrated before. So we see certain subtypes. This is usually the P and the I subtype, which do not have a lot of those neuroendocrine expression genes. And we have the small cell A and the N subtype, how we call it, which is a much more neuroendocrine gene. And the important thing is this, again, confirms to us that what we are looking at is there. It is reproducible. There's really biology that is going on and that can really help us to understand what is going on to really address the right method. However, the important thing that we have, and this goes back, like we're trying to be as fast as possible and find the most robust thing is, and I will tell you in a moment why this is important. RNA that we have here, so under this transcription, is something that is technically very nice. And we can look at this very robustly with a lot of different tools. But the important thing is RNA is a very, very unstable molecule. And like I told you before, we are very good at working with DNA because this is how we detect mutation. Because actually DNA is a very stable molecule. And the funny thing is they look very, very similar. We have this double-stranded DNA and we have the single-stranded RNA. They look very, very similar. But the important thing is DNA is just because of this double-stranded nature and something else so stable that we can look at, old animals that were, 20,000 years buried in ice. We understand about Egyptian mummies from DNA because it's a very stable molecule. While RNA, we don't have the possibility because of the way it is shaped, is unfortunately very, very unstable. And especially when we work with clinical specimen, we use certain methods to preserve the specimen. And those are very harsh to the environment. And actually what we see is that RNA, which usually has a certain length, it's like, a couple of thousand base pairs long, gets degraded to very, very small fragments. So while we are still very convinced that we can work with this RNA molecule, the first version of the classifier I just showed you, we still have the idea, can we achieve the same with DNA, where we have much more experience, where we know how we can do it and how this is actually working. So the interesting thing is, as we have this interplay between DNA and RNA, where we go from this genetic information in the end to a protein, there is a process which we call DNA methylation. So it is a very fundamental biological process in which actually the DNA is changed in a certain way. And when the DNA is changed in a certain way, then this part of the DNA can be read by certain proteins and then leading to the production of our RNA and our proteins. And the important thing is, as we look at the differences in the presence of certain proteins of our transcription factors, this is interestingly a very interesting thing because we now know that changes in the DNA have an impact on how many proteins we have. And now we have the idea that we can look at the DNA, which is much more stable to look at, and still get information on actually what is going on in the tumor itself. And therefore, the second idea was, can we actually build the same classifier that we had before, but now using DNA, which would things easier in routine clinical care, and I will also show you why this is extremely particularly exciting for another approach to really get our robust biomarkers, because again, we cannot get the right treatment when we don't have the right biomarker. And when we have the right biomarker, we can investigate the right treatment and then ultimately really shape how we can treat patients. And the important thing is that this DNA methylation is something that we already know in small cell lung cancer is very different. There have been already 7 years ago, there was a paper where they looked at DNA methylation. Also, looking at like how does it shape, and they found like 3 clusters which are very different in DNA methylation, which is very nice because I think about when we looked before, we found 4 clusters of small cell lung cancer subtypes. Now, like 7 years ago already in DNA methylation, we saw that there are very particular situations. We also see a lot of differences in DNA methylation between small cell lung cancer and non-small cell lung cancer. And all the time when you see something is very different, it usually means that there's something biologically which changes, which is very important. And there was even a nice paper. You see, we are speaking about 2020, and I will from now on only show you, new research which was published 2021 this year. And already there were some research which were able to show that certain of those DNA methylation changes are even already predictive of a therapy when we use cell lines in the laboratory. So this goes back, like, we know there's something going on, and now we have the possibility to really better understand the biology and really associate the right treatment. So what we then did is that we had a big cohort actually of small cell lung cancer samples. And what you can see here is actually all of the human chromosomes. It's a very high analysis. And we looked at the methylation level across basically the whole genome in a very, very low resolution. And the interesting thing is that already by looking at this, we see differences in those 4 subtypes. We see that, for example, in the small cell N, which is the green line, there's more methylation, while in small cell P, which is the red line, we see less methylation. And this is very, very critical because only when we see differences, then we can use them to build our biological classification system. This was very, very interesting for us because this means there seems to be changes, and hopefully we can now use them to define new biomarkers for this. We therefore then use again the same classification system as before. The idea was before the same, that we have the possibility to say, no, I don't want to bore anybody with machine learning. Even when I present it to my colleagues, they all start like, okay, come on, show me results. And the important thing is that we now actually get the same that we had before. So when we now compared our RNA-based classification to our DNA-based classification, we had an accuracy of over 98%. There was precisely one sample where we have seen, which was kind of misclassified. It was an RNA, it was a small cell N. In our DNA methylation, it was a small cell A. But we already know that there's a certain heterogeneity in those subtypes. So this was even still very, very important because we have this high accuracy. And critically, I showed you in the beginning that we have those samples that we couldn't classify from RNA because we had this not sufficient information. And 2 of those samples, we now suddenly were able to associate the right subtype, really highlighting it when we now probably, we don't know yet which is the right way to do it. So we develop everything in parallel. But it seems like with now this DNA methylation approach, we are really much, much better at getting information on more samples, which is ultimately our goal. The really interesting thing is also that when we then looked at the differences in survival, because we don't know, are there any differences? We want to get the same outcome. We could actually see that when we use our RNA-based classification or our DNA-based classification, we get the same outcome. So this really highlights that we have now 2 systems that we can use interchangeably, and we are really going in the right direction. The really important thing about DNA methylation, however, and this is super exciting, is that you might be familiar that we have something that we call circulating tumor DNA. So a lot of tumor DNA is shed into the bloodstream. And we use this quite often because this helps us to get a lot more information on the tumor and what is going on in the tumor itself by just simply taking a blood draw and not doing a very complicated analysis. We don't need to get information to the tumor. For example, in many of those targeted therapies in non-small cell lung cancer, we use this a lot because we can see, for example, resistance mechanism that we can then really define and change the treatment of it. Usually finding those circulating tumor DNA is very, very challenging because there's not a lot. So this is really where we had a lot of technical problems. How can we find it? The important thing in small cell lung cancer actually is the solid tumor where we have the highest amount of circulating tumor DNA. So this is a cancer type where we have access to a lot of those tumor-derived material from a simple blood draw. And this is really, really important for us, because when we get information from plasma, when we just take blood, then this makes a lot of things much, much easier for everybody. It is much, much more convenient. And now we are in the interesting situation that we have the small cell lung cancer where we really get access to those information and we can look at it. And the important thing is the DNA methylation. Those DNA changes that I've shown to you are important to define how we see the change of certain proteins are actually preserved in plasma. So the important thing is that we actually have the idea that we can get the same amount of information that we get from the tumor itself from a simple blood draw, and that we can then have the possibility to get those subtypes in something that we call a liquid biopsy approach, which is that just by taking a little bit of blood, we can get all the information that we need to get the right treatment, that we don't need biopsies, that we ensure that we also have enough material that we can really look at it and really get this into a situation where we can really impact clinical care taking. The important thing is, again, I told you there's a lot of things which are going on, and looking at those liquid biopsies on small cell lung cancer is something that have attracted a lot of speed this year. So there were 3 very important papers which came out just this year. We are speaking about the past month. One was really highlighting how you can detect small cell lung cancer from DNA methylation, how this works from a team from Benjamin Locks in Canada. There was a very, very important paper from Carolyn Diver, who's working in Manchester, where they actually already used those circulating free DNA to use it already for subtyping small cell lung cancer. They used a slightly different approach. So they had this ASCL1 predominant, this MURD1 predominant, and they shaped it in 3 different, not in 4 different subtypes, and naming the 3rd one double native. But they have already demonstrated that we can actually use this DNA methylation for the subtyping. And there was also another team from the NIH this year, which used another methodology where they looked at, we call them nucleosomes, who could also demonstrate that actually the information, how the biology of the small cell lung cancer is actually preserved, and we can really exploit it from liquid biopsy. So 3 important papers, with ours hopefully 4, just in 1 year, really highlight how much speed we have there and how the field is now rapidly changing. The important thing is we then also had the possibility to look at DNA methylation. We compared to tumor samples actually, and we could really highlight that the information that we get is really preserved in plasma. For example, what we had here is we were really looking again over the whole genome. What is the DNA methylation level in the tumor sample? What is the DNA methylation level in the plasma sample? And you can see, I need to speed up, I think a little bit to speed, so I'm very sorry, that everything goes around the 0% line. So we get exactly the same amount of information from the tumor from the plasma that we get from the tumor. And we then actually use this and applied our classification system and could really highlight that when we're now using our plasma samples, that we are also capable. There's again 1 sample which was a little bit off. Now we have a much bigger data set and we have this much more robust, but we can really now do the subtype classification in small cell lung cancer from plasma samples. And the second important thing is, and this is my last slide, why this is important, because the important thing when we look at blood is also that we cannot only look at what is happening, for example, at the moment of diagnosis, but it helps us to understand what is happening in the tumor over time. And especially the important thing is when we have those subtypes, we are exploiting them to find a new therapeutic approach. And in the moment we are doing it, it can happen that the tumor, for example, changes its subtype to await the treatment. So when we have something like plasma, we can even track this over time and have the possibility to, for example, imagine when you have a subtype change, then we can give another treatment. So we can really try with this new liquid biopsy approach to go with this over time. And actually, we had 2 patients where we had the possibility to get those blood samples over time during the treatment. And the important thing is we had the first patient. And for the first patient, when we looked at the tumor of this patient and the plasma that we had there, from the beginning, all over the treatment, the tumor were in the small cell A. Nothing changes there. So we really saw that there was no changes for this tumor. So again, in the distant future, when we could really demonstrate that we can use those to find the right appropriate treatment, you could say like, hey, this patient should stay on the appropriate treatment because we are not seeing this. However, for the second patient, where we also had the possibility to look at longitudinal samples, we could see that initially this tumor was a small cell A, as we would have classified this. But over time, over treatment, we could actually see that it went into something, this equivocal state, where we were not even able to say which is the subtype. So you could clearly see that something was going on in the tumor. And then actually showing a switch of the tumor to another subtype, which is highlighting that those subtypes are unfortunately not that stable. But when we have those plasma samples, we are really capable to track this over time, that we can really be this step ahead. And if there are changes, that we can adopt this, the treatment, find new ways, new treatments to really try to be, always this one step ahead. And this is really the great idea when we now use this DNA methylation, that we have this preservation there. And this is actually the talk. I'm so excited I had the possibility to speak today. I hope you see it as a really rapidly field. I hope probably by the moment I stop this talk, there are already changes. The important thing is that now we have those small cell lung cancer subtypes. We have discussed that they are prognostic, but we are working to getting them predictive. And really, we have the possibility, especially with this DNA methylation, to subtype those small cell lung cancer samples in a clinical context, hopefully get this tracking over time. And very importantly, there are many people involved. This is started as a very small project. We have now half of the world, I have the impression, which is really helping us. All this research is driven and led by John Heymach, Lauren Byers, Carl Gay, Ignacio Stuber from Medical Oncology, from Pathology. We have a lot of collaborations partners from all over the world. We have a lot of funding sources which are really there to help us to do this research. It is really very important for all the support of our patients, really enable us to do this research. The important thing is small cell lung cancer is a really team field, and we were very excited. Actually, last week we were nominated even by the MD Anderson for the Team Science Award. But I think it is also very important to highlight that we can only do it all together when we're all working together. It's really the only possibility that we find something that is going on there. And then as a very last slide, I know it's the 19th of December. I take the opportunity to already wish you and all your families already happy holidays. I hope you can really appreciate it, and already all my very best wishes for next year. And I'm really excited about all your questions. And again, thank you so much for the invitation today.
Wow, thank you. I did not know that liquid biopsies were kind of standard moving for small cell. That's great. But we have another question in the chat. Is it, let me pronounce it correctly, atezolizumab, or otherwise known as Tecentriq, is that still available?
What do you mean with available? So this is still approved by the FDA.
Okay, another question. Are the liquid biopsies already standard of care?
So yes and no. So liquid biopsies are standard of care in non-small cell lung cancer. Mm-hmm. And in actually many other cancer samples. So it is still that, for example, there are still debates, so to say, but for example, when there's no possibility to test the tumor of a patient, it is absolutely standard of care that you do a liquid biopsy. We had a discussion before about those targeted therapies, about those TKIs. Liquid biopsies are absolutely standard of care to track for resistance mechanism since a couple of years right now, to really track also tumor evolution over time. So they are definitely standard of care in many cancer types. They're not standard of care in small cell lung cancer for the sheer reason that they are standard of care to guide a certain treatment. In small cell lung cancer, we don't have those treatments where we need the guidance of those liquid biopsies. There's a lot of research which is not yet clinical. For example, that you saw that the tumors are shedding DNA in the bloodstream. So the idea is, for example, when you have more DNA in the bloodstream, that you have more tumor. And the inverse idea is when you see that you have no more DNA in the bloodstream, for example, that the tumor is much smaller. So there are ways that is not yet standard of care, but is occasionally done, that people look at this and use this to track a little bit the tumor size, so to say. But it's not standard of care to guide treatments because we don't have those treatments. What we're obviously hoping is with those subtypes we develop the treatments. And with the treatments, we need the methodology to follow those treatments over time and actually make it standard of care. And due to some of those particularities in small cell lung cancer, I'm very optimistic that this is going to happen, because liquid biopsies were usually technical challenges that we had because it was very rare. It was really just a technical problem. Everybody knew how to use it, but we had problems to define it technically. Now in small cell lung cancer, many of those technical challenges are either solved because of non-small cell lung cancer, or because there's so much more circulating free tumor DNA that we can really look at this. So again, I'm only presenting research today, but we are giving our very best that in a future meeting, you will talk about this as standard of care.
No, and we appreciate the research. It wasn't until 2019 that Tecentriq was even approved. So we're seeing the needle move. Does anyone else? I don't know if Dusty had any private questions in her. Oh, here, I do have a hand raised.
Thank you, Dr. Heeke. It's very interesting. In terms of immunotherapy not working, like atezolizumab for an extensive stage small cell lung cancer patient, would you think that that's because it was kind of like a shot in the dark and maybe their type is not going to respond to the PD-L1 inhibitor? Would you then make a supposition that maybe he had another subtype that this was not useful against?
That's a challenging question because there are many ideas, many theories, but we are still trying to understand it. So the important thing, those subtypes are one way we are trying to solve this. So the idea is, it seems like, when the immunotherapy, for example, is not working, there's some biology which are the reason that this is not working. One of the ideas that we had is this inflamed subtype, because we know that there's a different immune state. And obviously when the immune system is a little bit different, and when we then target the immune system, we have higher chances. The important thing is the subtypes are very, very strong simplification of a very complex biological phenomenon, and we are yet to really understand what is going on there. So we hope that the small cell lung cancer subtypes contribute to it, and maybe it was what we now would say a different subtypes, but there are so complex reasons why this is working, why this is not working, that the simple answer is, at least I don't know yet. So we really try to understand more, just to understand the biology.
Anyone else have any questions? Great information.
I just wanted to thank you, Dr. Heeke, because you are so enthusiastic that you give us hope. Not all oncologists are enthusiastic, and you're the first actual researcher I've ever listened to. So thank you for just being excited about trying to help us.
Thank you so much. I can tell you we are really giving our best, because we know why we do it, and keeping enthusiasm is a very important. But to be honest, I think things are moving now, and this is the important message. Everything that I showed you today is not yet in the clinic, but the message is things are moving, and we are very, very optimistic that there will be changes, and we're really hoping that we get this. But really, thank you very much. And again, I cannot stress out how much I appreciate having the possibility to speak to you today, because that's what we are working for.
And you might know the answer to this, you might not. Are there any active clinical trials for small cell lung cancer patients out of MD Anderson that your colleagues are conducting?
Oh, we have many small cell lung cancer clinical trials in all different settings: first line, second line, third line. Many of them are led by the medical researchers, by the medical doctors that I mentioned before: Dr. Lauren Byers, Dr. Carl Gay. We have Dr. Marcelo Negrão. There are many of those. I might have missed now some, so I'm very sorry for those which we have missed. No, we are extremely active with clinical trial. Actually, the MD Anderson is still, I think, one of the largest clinical medical cancer centers in the world. So we are seeing a lot of patients, which is very important that we really have those clinical trials running, and we hope to have much more in the future. So yes. Definitely, yes.
Awesome. Anyone else have any questions? Thank you so much what people are saying in the chat, and we are very grateful to you for coming here and sharing your information.
Isn't that a hand up? It's clapping.
Oh,
thank you so much. Thank you.
Yeah, good. Thank you so much.
So I think the message that you are leaving with us is that the needle is moving, and that there's hope on the horizon.
Yes.
Yeah. Thank you. We hope that you enjoy your evening.
Thank you so much for taking the time to give us that. There's a lot to unpack, but we can taste the hope, and it's exciting. We're excited too. So you've encouraged us. Thank you so much.
And well, I have a question. What made you interested in studying small cell lung cancer?
I was thinking about this question a lot, and the weird thing is I don't even know how I came into lung cancer, to be really honest. I think what usually makes you interested is meeting people who are enthusiastic and makes you excited about something, to be really honest. And I think that just, I just met people who were excited in lung cancer, and I was like, oh my God, this guy is excited now. I want to do the same, for example. Maybe if I would have had somebody in colorectal cancer, I would now do colorectal cancer. What made me interested in small cell lung cancer was this issue that there was not enough work done in my personal, well, maybe there was enough work done, but we had so much good movements in non-small cell lung cancer, and there's still this big field with just, where we needed to find solutions and then build it on this. I started this, I joined the MD Anderson 3 years ago. At the time, really everything moved, and we really figured out that something is going on. And it's just exciting now in a time where you really see we are onto something. We can make this difference. You don't spend, 15 years in the lab hoping to find something, but I'm now in a position where I can make an impact and change something. And probably this is why I have this enthusiasm, because it's like, now is the right time. We had sometimes people calling it the second golden age of small cell lung cancer research, to really all the research is really now moving to something where we have the hope to find something that can really make an impact. And I think this is the best motivation that anybody can have for working.
Well, definitely. Thank you for the work that you do. We do not want to be the Cinderella child of the lung cancer world anymore, or the cancer world for that matter. And are the forget-me-nots. So thank you for the work that you do.
Thank you very much. I wish you all happy holidays. It was really a great honor being here today. Thank you very much. Goodbye.